Good morning, everyone. We are going to start a new chapter today, but I have one question and a couple of slides that I want to reiterate from our previous chapter on um, genome evolution. So we will start with that for a couple of slides, and then we'll move on to chapter 11. So if you want to get the previous PowerPoint out from where we left off on Monday. Okay, there we go. So looks like we've got some people still arriving. We are going to start a new chapter today. We're going to be looking at our membranes, what our biologic membranes are made of, and what kinds of roles they have in the living cell. I want to remind everyone that Joshua has his office hour. His regular office hours are Thursdays at 11. And right before each exam, he'll have that additional Tuesday afternoon office hour that I'll remind you of. But his regular office hours are Thursdays at 11. And I have my office hour, my Zoom office hour is today at 1 o'clock, in addition to my chat office hours that I have Mondays at 12:30. So is everybody ready to roll today? All right, I wanted to reiterate or repeat the last couple of slides that we had covered on Monday in our previous chapter looking at genetic changes, because a couple of people had a couple of questions for me, so I feel like maybe I didn't give it a thorough enough explanation. Our last type of genetic change that can contribute to the evolution of a species is what we call horizontal transfer. And we described one example of how a piece of genetic information, a gene, a segment of a genome, can be transmitted from one organism to another. And one of those processes that occurs in bacteria is conjugation. And we showed a slide of that, of a relatively common natural phenomenon of one bacteria communicating with another bacteria. And this transfer can be between species as well, via this sex pilus, which is stained with an antibody. That's why it looks so large relative to the microorganism, where information from one individual, one cell, can be transferred to another cell. And again, this is one way that bacteria can pick up antibiotic resistance genes carried on plasmids, and be able to acquire that resistance. This we call conjugation, and that is among microorganisms, among bacteria. And again, I mentioned that E. coli, a common bacteria, has acquired about 20% of its genome from other bacterial species over millions of years. About 20% of that genome has been acquired via this type of conjugation. But also remember, there's another way for genetic information to be transferred from one organism to another, and that could be via viral infections. Um, a virus that is present in one organism, when it replicates and leaves its first cell and then infects another cell, may actually take some genetic information from its first host and bring that to another host. That's another method or mechanism for horizontal transfer of genetic information. So I have a clicker question for us today. Our first question, make sure that you have logged into your Reef account and you have joined our course. Our first question today is sort of a summary question from the information in our previous PowerPoint. Questions about genome evolution. So here we go. I've got 5 statements listed here, and your job is to identify which one is true. So find something wrong with 4 of them so that you can identify which is the correct statement. How's everybody doing? Can you identify something wrong in each of 4 of the statements? All right, let me close it up. 5 more seconds. Everyone had a chance to vote. All right. The one statement that is correct would be C. And C states that a pseudogene is similar to a functional gene. It's usually part of a gene family, but it can't be expressed anymore because it's acquired mutations over time. So we call it pseudo. The other ones are incorrect because evolution is not building from scratch, right? It's going to nature is selecting for functions that are advantageous to an organism, and doesn't necessarily always come up with the best possible solution, right? So evolution is constantly changing, but not making completely new pathways. So A is not correct. B is not correct because not every instance of a DNA duplication leads to a new functional gene. Sometimes that duplication leads to a pseudogene. Multi-gene families are very common in vertebrates, right? A large proportion of vertebrate genes are part of gene families, such as different types of motor proteins like myosins and kinesins are parts of large families. Immunoglobulins arose by gene duplication. Let's see, different types of G protein coupled receptors. We'll talk about those in a later chapter. The multiple different types of those arose by gene duplications are members of multi-gene families. And as we just stated on our previous slide, this horizontal transfer is very common in bacteria and has had a big impact on their genomes. Okay. All right, so now we're going to move away from our chromatin, move away from the function of the components in the nucleus, and we're going to look to the surface of our cell. Oh, before I go there, yes, the what you should know. I always will give you a list, sort of a brief summary of some of the questions you should be asking yourself to make sure that you understand the main concepts that we've just discussed in that chapter. So now we're ready to move on to our next chapter. It's PowerPoint 8 and covering material from chapter 11 and 12. And we're going to look at our biologic membrane. First, look at the structure, the components that make up the membrane, and then we will look at one of its very important roles in regulating transport, regulating which compounds, which ions are able to enter or exit the cell. So if we look at the structure first, the basic component of our biologic membrane is that lipid bilayer, 2 layers of our amphipathic phospholipids. Amphipathic meaning that one end is hydrophobic, that is excluded from water, and the other end is hydrophilic, that it interacts with water. And because these phospholipids that make up our biologic membrane are amphipathic, they will spontaneously form this bilayer, right? All of the hydrophobic fatty acid tails will be excluded from water and will end up essentially protected from the aqueous outside of the cell and the aqueous cytoplasm of the cell. And this layer will spontaneously form a sphere as well. The sphere to protect those hydrophobic fatty acids from the water, right? The best way to protect them from water is to have a continuous sphere of this bilayer. So if we look at our phospholipid in a little bit more detail, remind ourselves, we talked about it in earlier chapter. Our glycerol, 3-carbon glycerol, is bound, 2 of the carbons of that glycerol are bound to fatty acids. These are these hydrocarbon chains. Usually and there's different types of phospholipids, but most of the time, one of those fatty acids is a saturated fatty acid, and the other fatty acid is a cis unsaturated fatty acid. So it has that little kink in it. And that's going to be important for the function of this lipid bilayer that we'll talk about in just a minute. Again, our glycerol is also, that 3rd carbon is linked to a phosphate group, which is linked to one of many different types of polar groups, which makes this end of the molecule hydrophilic. It will interact with aqueous solution. So it is exposed to the water on the outside of the cell, and on this side of this lipid bilayer, that the polar group of that hydrophilic head is exposed to the cytoplasm, if this was the inside of the cell. Now, one reason for the presence of these different types of fatty acids in a phospholipid is to make sure that this lipid bilayer is not too rigid and it's not too fluid, right? The presence of the little kink in there makes sure that these phospholipids don't pack together too tightly, but the presence of this rigid saturated fatty acid prevents these phospholipids from being able to move too much, from being too fluid. And our lipid bilayer is actually in animal cells particularly, is actually has the fluidity of oil, salad oil. And we'll talk about that in just a couple minutes. So fluidity means that the molecules are moving relative to each other, constant molecular motion. And the movement of these phospholipids is actually fairly rapid diffusion, what we call lateral diffusion. Within a given layer, these phospholipids are constantly exchanging place with each other. They're moving about 1 million times per second. They can flex, they can rotate, they can move laterally, right, within one of the 2 lipid layers. What they almost never do is switch from one layer to another. And we can see why that would be very difficult. That would be thermodynamically difficult thing to do because we've got this hydrophilic head that's going to have a very hard time passing through this hydrophobic region of the bilayer to get to the other side. And so when that does happen, it would have to be something active. It would have to be something that would be enzymatically catalyzed to actually move a phospholipid from one layer to the other. And we'll see an example of that in a later chapter. Why a cell would choose to deliberately do that. So again, most of the time, the lipids, the phospholipids that are in one layer stay in that layer, but they can move around. The lipids that are in the cytoplasmic layer facing the cytoplasm stay in that layer as well, but they're moving relative to each other all the time. Now, we also saw in our first slide, but I forgot to point it out, that we, of course, have proteins within that lipid bilayer. And so our next slide is answering the question, do the proteins move within those lipid bilayers as well? And this type of experiment uses antibodies to label the proteins in different cells. So what we have here is an experiment carried out in cell culture that a mouse cell is labeled with an antibody that has attached to it a rhodamine compound that fluoresces red underneath a fluorescent microscope. And they have then also labeled a culture of human cells with an antibody that's attached to a fluorescein molecule, which will fluoresce green. So we can distinguish the proteins that are specific to an animal cell, distinguish them from the proteins that are specific to a mouse cell. And then you can experimentally induce those cells to fuse. That can be done chemically, that can be done by applying an electric shock, which will cause the two bilayers to actually fuse with each other. And we end up with this hybrid cell. And what we see upon fusion, that we have all of the labeled proteins that are restricted to the particular half of this hybrid cell from which they arose. But if you allow the cell to be incubated within the right kind of medium for about an hour, what you see is that all of those labels are now spread throughout this fused cell, meaning that all of those green and red proteins that originated from one half of the cell have now diffused. They have now moved within the lipid bilayer and sort of spread out amongst that cell. So as well as the phospholipids moving within the lipid bilayer, within one of the layers of the lipid bilayer, the proteins that are embedded within that lipid bilayer can move as well. It can move laterally, and that's going to be important. If you think about, for example, if this wasn't an experiment but was a fusion of two gametes, a sperm cell and an egg cell, right? When they will be bringing different proteins to that fused cell of that diploid zygote, and all of those proteins have to be able to spread throughout that zygote. If you think of a cell is dividing, it has to be able to distribute all of its proteins to the two daughter cells upon division. So that's going to be an important feature of the function of these proteins, as well as the phospholipids in a membrane. So let's sticking with the lipid layer for a little bit. Let's look at it in a little bit more detail. In animal cells, that lipid bilayer also has about 20% of the lipids are cholesterol. Cholesterol is a steroid-based compound, and all steroid-based compounds have this rigid planar structure that is actually 4 fused rings. This is all nonpolar, and a 6-carbon nonpolar hydrocarbon tail extends from one end of this steroid ring, and at the other end is a polar group, right, a hydroxyl group. This cholesterol molecule will insert itself in that lipid bilayer, sort of in between two phospholipids, with its nonpolar group extending into the nonpolar region of the membrane, and that polar sort of head group exposed to the water. If we look at a lipid bilayer, we see about 20% of all of the lipids are these cholesterol molecules that have been inserted into the layer. Now, the cholesterol itself can't form a bilayer, but it can insert itself into bilayers that form by the assembly of different phospholipids. So why do animal cells have cholesterol, and why is it an important feature of animal membranes? Is because cholesterol can help modulate the fluidity of that membrane. We want that the membrane has to be able to have its constituent parts move around, but it can't be completely fluid because it has a role in the cell, right? It has to actually serve as the boundary between the unit of the cell and the environment that the cell finds itself in. And so in animal cells, the role of cholesterol is to help make sure that it can maintain the stability of that lipid bilayer, that it doesn't become too fluid. So that presence of cholesterol is going to restrain the movement of the fatty acids, so they're not moving too much. But then at lower temperatures, the presence of cholesterol prevents the fatty acids from packing too tightly. So it helps maintain some degree of fluidity. So it's serving sort of as a temperature buffer, preventing too much rigidity of the membrane at low temperatures, preventing too much fluidity at higher temperatures.
Okay, I found this on.
Sorry, X is trying to talk to me. Okay, so that's our lipid bilayers. And looking at our lipids again, we can also see that there's a difference among those bilayers as to what kinds of phospholipids we actually find in those layers. Asymmetric distribution of the actual phospholipid components. The outer layer of the membrane, meaning the layer that's facing the extracellular environment, consists of phospholipids that have sugars added to them. Glyco meaning sugars added to the lipids. The side that's facing the cytoplasm, the inner layer of the membrane, is where you will find certain types of charged phospholipids, like phosphatidylinositol, phosphatidylserine. And the reason that those are on the inside is that those, let's label them here first, and I'll tell you the reason for both of these. Glycolipids, here our little symbol for this little six-sided sugar ring, we find them on the outside, facing the extracellular space. In contrast, some of these phospholipids on the cytoplasmic side are these charged phospholipids. And we'll see in later chapters when we're looking at signal transduction that some of those phospholipids, these charged molecules that have particular groups attached to them, will actually interact with cytoplasmic components and change the behavior of the cell. So we have these sugars on the outside. So glyco again, meaning a sugar component added to a lipid. And one of the roles of these sugars is to give an identity to the cell. So different cell types will have different sugars on their surface. Different types among different species, among different cell types, among different tissues in an individual. Different individuals can have different carbohydrate markers on their surface, right? It's one way for cells to be able to identify each other on the basis of the sugar components that are on the surface. And then again, notice what we've shown in our lipid bilayer is the frequency of the presence of those cholesterol molecules, right, inserted within each of those layers. All right, so that's our lipid bilayer. Phospholipids plus some cholesterol in animal cells, but a variety of different types of phospholipids that have different roles in a cell. But a membrane is not just the lipid components. Actually, the components that really do a lot of the jobs of the cell are the protein components. And the protein components, the main ones that we'll be spending most of our time talking about, are transmembrane proteins. So here we have, and we'll be using this symbol in many of our images, where we have the sort of two gray bars are representing our lipid bilayer. And all of these protein components that we're looking at this point are transmembrane proteins, meaning that these are proteins that are embedded within that lipid bilayer. We can divide them into different categories. And we'll be talking about all of these in different chapters when we're looking at different processes and different functions of cells. Some of these are transporters. We'll talk about those today. These are proteins that that span the membrane, and they can either form a channel that allows components to be able to move across the membrane. They may form what we call carrier proteins that can actually move or direct movement of solutes across the membrane, one direction or another. Other proteins, like these blue ones here, serve as anchors. They may bind the cytoskeleton, like actin filaments inside the cell, and may bind some extracellular matrix proteins like collagen on the outside of the cell. May help to anchor components of the extracellular matrix to components of the cytoskeleton inside. We'll talk a little bit about some of those in later chapters as well. Some of these transmembrane proteins serve as a receptor. They may bind a signal, such as a hormone, that is present in the extracellular space. And by binding to that signal, will cause a change in the shape of this receptor protein that will activate a particular type of biochemical process inside the cell. So receiving information outside the cell, relaying that information that is going to affect the behavior of the biochemistry inside the cell. And some of these transmembrane proteins have catalytic activity. For example, a tyrosine kinase present in the membrane can catalyze a specific chemical reaction, such as transferring a phosphate group in the case of a kinase. And again, we'll see examples of all of those later. If we look at our proteins in our lipid bilayer, again, we want to think about them in a couple of contexts. The ones we've talked about so far, we can call integral membranes because they have a transmembrane domain. So for example, again, here's our lipid bilayer, our two large gray bars, and all of these green symbols are proteins that have a transmembrane domain that are spanning that lipid bilayer, right? They have a part that's exposed to the extracellular portion, a part exposed to the cytoplasm. They're embedded within that lipid, the lipid bilayer. Integral proteins are proteins that are embedded, right, and span the membrane. Integral transmembrane proteins. Other proteins are associated with the membrane, right? They don't actually span the lipid layer like this here, but so we call them peripheral proteins. They are bound to, they're forming a chemical attraction, chemical bonds with an integral protein. Right? So peripheral proteins are associated with the membrane. We usually find them on the extracellular side, and they will be interacting with integral proteins. Now, what we also notice is that all of these proteins on their extracellular side have sugars attached to them. So these are all what we call glycoproteins, right? So just like we said, many of the phospholipids of the lipid bilayer are glycosylated, meaning they have sugars added, so they are glycoproteins. Many, if not all, of the proteins in the membrane have sugars added to the outside. So if there's just a few sugars, we call it a glycoprotein. If we have really long polymers of sugars, polysaccharides, then we call it a proteoglycan. But it's pretty much the same principle. Part of the reason for this carbohydrate, or one of the roles of all of these carbohydrates that we call a carbohydrate layer, is to protect the cell. It protects because it's covering the whole cell surface, it's going to protect the cell from damage, from chemical damage, from mechanical damage. In addition to, as I had mentioned previously, the presence of different combinations of sugar subunits on these glycoproteins and glycolipids will give a specific identity to a cell. So that allows cells to recognize each other, to be able to bind to each other, to adhere to each other, to be able to form a complex of proteins to form a particular tissue, for cells to recognize that they need to associate or assemble with each other. All right, so we have our lipid components of our biologic membrane. We have our protein components of the membrane, and on the outside, we have this carbohydrate layer, these sugar components that are attached to either the proteins or to the lipids. So what have we talked about so far in the context of our proteins of our biological membrane? Which one of these statements is true? Do plants have glycoproteins? Well, the cell wall is primarily carbohydrate. So cellulose is carbohydrates, and there are some proteins in there. And that cell wall is attached to the bilayer. So there would be part of the cell wall and part of the membrane would have sugars associated with it. I'm answering a question in the chat. So think about what's wrong with the other statements, right? What's wrong with the 3 that you're not choosing as true? How about 5 more seconds? Everybody had a chance to vote? All right, let me close it up. And the one statement that is true would be B. The protein that relies on protein-protein interactions is a peripheral membrane protein. Transmembrane proteins are what we call integral proteins, right? Because they do pass the bilayer. So A is not correct. A protein that binds to the cytosolic side would not actually, if it did bind to the cytosolic side, it would be interacting with the protein, not the membrane bilayer itself. It would be interacting with the protein, and it would probably be interacting through hydrophilic side chains, not hydrophobic side chains. And the group of membrane proteins that serve as channels or carrier proteins that move ions, or in the case of ions would be channel proteins, we call those transporters, not enzymes. So what we want to spend the rest of our time, now that we have defined some of the components of our membrane, we want to actually look at what's the membrane doing. And what's one of the things the membrane's doing, really. Here we're going to be talking about the membrane regulating what type of molecule or ion is crossing the membrane, right? What substances are coming into a cell, what substances are leaving a cell. Now, if we look at just a lipid bilayer, right, and here we have our 2 parallel bars of just a lipid bilayer with no proteins in it, what we would find is that lipid bilayer would allow small hydrophobic molecules to be able to pass from one side to the other. And they would be moving, they would be able to diffuse across, they would be moving down their concentration gradient. So very small molecules like gases, oxygen, carbon dioxide, small molecules would be able to pass one direction or the other. Even polar molecules, as long as they're not charged, and if they're small enough, like water, should be able to, maybe a little bit more slowly, right? Water more slowly than oxygen or carbon dioxide, but they can um, interact with the hydrophobic components of the lipid bilayer and can move across, right? From a region of high concentration to low concentration, they can diffuse across. But a polar molecule that is larger, like an amino acid or a sugar subunit like glucose, they're too big to be able to dissolve in that lipid bilayer and they can't pass across. And ions, right? Ions, because of their charge, cannot cross a lipid bilayer. So the charge, the size, the attraction to water is going to determine whether or not, right, something can cross a lipid bilayer. And so that's one of the roles of our biologic membranes, is to be selective as to what can pass, right? Which kinds of nutrients can enter a cell, what wastes can leave a cell, right? So the bilayer is going to selectively determine what's being transported across the membrane. Now, this is where the proteins come in, right? Proteins will be required for large molecules, charged molecules, charged ions to be able to cross that biologic membrane. So our biologic membrane has proteins embedded in that lipid bilayer, and those proteins we call transporters. So that's what we're going to be looking at today. When we're talking about transport or movement across the membrane, the first thing we're looking at is whether that's going to require energy or whether it is a spontaneous process. And the first types of molecules that we're looking at, for example, looking at oxygen, carbon dioxide, those small gases, small non-polar molecules that can cross the lipid bilayer, they can cross freely, right? They can move down their concentration gradient. And we call that process diffusion, right? Moving from a region of high concentration to across the membrane to a region of lower concentration, right? Diffusion is the tendency of a solute to essentially fill the space, right? Because of the constant molecular motion, just by moving around, eventually there would be more solute moving from one side of the membrane to the other side, down its concentration gradient, right? And again, we call that a spontaneous process. An example of diffusion, simple diffusion, and this is a passive type of transport as opposed to active. Passive because it's spontaneous and no energy is required, would be osmosis, right? Osmosis is the diffusion of water molecules specifically. And if we look at a cell here, we would see that, and we're comparing what is the direction of water diffusing across this cell's membrane. Note that, of course, a cell has a lot of solutes in it, a lot of macromolecules, a lot of ions, right? A lot of membrane-bound components. So the cell has, even though its cytoplasm is aqueous, right? Everything's dissolved in water. There is relatively less water. In this example, we're looking at a cell that has a high concentration of solute inside the cell compared to, and our little red dots are our solute, compared to if we put this cell in a solution that would be hypotonic, where we have a low concentration of solute outside the cell. What that would mean is that there's more water outside, higher concentration of water outside than inside, which means that the water, the net movement of water would be into the cell. That's going to cause the cell to swell, right? Water will diffuse into the cell. The net movement of water would be from outside to inside until the two concentrations we reach equilibrium, right? Which would eventually only occur once the cell burst, which is why it's very important that our cells our biological cells are in an isotonic solution. Our cells have a very similar concentration of water inside the cell as the extracellular fluids that they are floating around in, to make sure that we have what we call an osmotic balance, so that overall, every molecule of water that diffuses in, another molecule is diffusing out. The rate of diffusion into the cell equals the rate of diffusion outside the cell. Therefore, the cell doesn't shrink, the cell doesn't swell. And that's why all of our cells are, our bodily fluids, our cells are floating around in bodily fluids that are similar in ionic strength, similar in solute concentration. Outside is similar to the concentrations inside. And our bodily fluids are pretty much the saltiness of seawater, a high concentration of sodium ions, high concentration of chloride ions, and that helps to balance the concentration of solutes inside the cell to make sure that our cells have an osmotic balance. The net influx of water into the cell equals the net efflux of water outside of the cell. So diffusion is movement of a solute from high concentration to low concentration. That's our simple diffusion. Now, there are some solutes that can't move across that lipid bilayer because they're either too large, because they're polar, or because they're charged, but they still can move across the membrane if they are facilitated in that movement by some proteins in the membrane. And so this is another type of passive transport that we call facilitated diffusion. The movement of these types of molecules, polar, large, charged, depends on transporter proteins. There's two main types of transporter proteins, either carrier proteins or channel proteins. And they allow a molecule or an ion to move down its concentration gradient. Again, passive, no energy required. The net movement of that molecule or that ion is from high concentration to low concentration. It's moving down its concentration gradient. And then, so we're going to look at a couple of examples of these transporter proteins. What's the difference between them? And then we'll be talking about how some of them actually work in a cell. So here we're looking at a carrier protein. And again, here are two little gray lines. That's our lipid bilayer. And here in green is our carrier protein sitting, right? So it would be a transmembrane protein sitting in that bilayer. And a carrier protein generally has a couple of different conformations, a couple of different shapes. It has a binding site that is specific for a particular solute. Our solute is in yellow. And that solute can bind. And because the carrier protein sort of arbitrarily changes its shape, the solute can move from one side to the other. And depending on, let's say that there's more of this yellow solute on this side of the membrane, eventually the net movement of the solute would be from high concentration to low concentration. So carrier proteins change their shape because they can bind to a specific solute. We contrast that to a channel protein, which again is another transmembrane protein, but it doesn't change its shape. That channel protein crosses the lipid bilayer multiple times and forms essentially a hydrophilic channel that allows ions to be able to diffuse through that space. So it's creating a hydrophilic pathway within the hydrophobic lipid bilayer that allows ions to move down their concentration gradient. So both of these types of transport proteins facilitate diffusion of a solute. right? Down its concentration gradient. Doesn't matter if it's going inside to outside or outside to inside, it's going down its concentration gradient. We compare that to that type of passive transport to, oh, I'll tell you in a second, to active transport. But let's think about for just a minute. If we're looking at these different types of solutes, which of these would be least likely to cross a synthetic lipid bilayer? A synthetic layer of lipids in which we do not have any protein components. We don't have any transport proteins. I see all kinds of things going by in the chat, but I can't tell if any of those things are, you guys seem to be having some extraneous conversations going on. So I can't read it fast enough to see if there's actually any questions going on there. I see a hand raised. Do you have a question for me? Daritza, do you have a question? Okay, if you don't have a question, can you put your hand down? Okay, so which of these, identify which one do you think is going to have the hardest time or impossible to actually cross a lipid bilayer? Everybody okay? Had a chance to vote? All right, let me close it up. And there actually are two possible answers. Both A and D are correct. A, glucose would not be able to cross a lipid bilayer because it's polar and it's too large. So glucose is going to require a transport protein, a carrier protein. The other one that's going to have a hard time to cross a lipid bilayer or will not be able to would be a potassium ion because of its charge. It's going to require an ion channel to cross the membrane, right? As opposed to carbon dioxide, small gas, non-polar, can cross that lipid bilayer. Water can, even though it's polar, it's small enough that it can. And glycerol, again, small enough that it can cross a lipid bilayer. All right, so this is an example. There were two possible answers: glucose, potassium. Neither of those can cross without assistance, without being facilitated by a transport protein. So everything we've talked about so far is passive transport, right? Movement of a solute across the membrane down its concentration gradient. But sometimes a cell needs to move solutes across its membrane against its concentration gradient. It has to push this solutes across. And this type of transport is going to require energy in the form of ATP. And the type of carrier proteins that can push something across a membrane are called pumps. And we'll talk about examples of active transport, of the function of a pump type of transport protein a little bit later. So let's put all of these together, right? Sort of a little summary. Solutes can cross a biologic membrane in a number of different ways. Passive transport requires no energy. Active transport does require energy. If we look at these different solutes in this example here, we see that on this side of the membrane, the top, there's a higher concentration of all of these than there are on the lower side of the membrane. And if we look at the little red dot, that would be an example of, let's say, an oxygen molecule that, because it's small and nonpolar, can pass from the side of high concentration to low concentration by simple diffusion. If we look at the little yellow ball, it can move from one side to the other from high concentration to low concentration, but it can't go through the lipid bilayer, but it can pass through a channel protein. Right? Again, passive. If we're looking at our larger blue ball, let's say that's a glucose subunit, glucose ring, that can move from high concentration to low concentration by binding to specific regions of its carrier protein. And then if we look at our green square, that green square can be pushed across from the region of low concentration to high concentration. That requires energy. That can be pushed across by a special type of carrier protein. It binds to it specifically, but it's moving against its concentration gradient, right? So we call that type of transport active transport. All right, so let's look at some of these membrane proteins that we have drawn here in our membrane. Right, we have channel proteins. We have some types of channel proteins that are gated, meaning that they're closed until the cell receives some type of stimulus that will cause them to open. So they're normally closed, but they can be opened. And then we have our carrier proteins. Right? All of those can facilitate diffusion of solutes. And then a little bit later, we'll look again at our pumps. These are transporters that require energy to drive a solute across the membrane against its concentration gradient. So a channel protein. A channel protein is a protein, transmembrane protein, forms a little pore, a hydrophilic pore that allows an ion to move down their concentration gradient. No energy is required. No changes in this in this channel protein occur. It just forms this little pore or channel that the ions can pass through. Contrast that to a carrier protein, such as a glucose transporter, where the solute, here's our solute, our glucose. Notice we have a higher concentration on the top portion, let's say that's the outside of the cell. It can just bind to a very specific binding site. And because this protein has essentially two different conformation states, two different shapes to it, the movement of the glucose is going to depend on its concentration gradient. If there's more glucose on the outside of the cell, then the interaction of glucose with its carrier protein will be to overall move glucose from outside to inside down its concentration gradient. If it turns out there's more glucose inside the cell, then the glucose will be able to bind to its carrier and its net movement would be to the outside. Right? What's important here is in both cases of the ion channel and the carrier protein, what the driving force in directing which direction the solute moves from inside to outside or outside to inside is going to depend on the concentration gradient. So we have to stop here because we have run out of time, but we'll pick up here on Friday and discuss what happens if the solute that's being moved has a charge to it. That sort of changes everything. So we'll pick up there on Friday, continue talking about transport across membranes. I do have my office hours today from 1 to 2. If anyone has any questions, they want to come join me in Zoom. Otherwise, you can talk with Joshua on Thursday or send me an email if you have any particular questions.
Professor, can I ask you something really quick right now?
Yes.
Okay.
Sure.
I have a question about back to the slide that was about the phospholipids and the proteins, how they can move around.
Mm-hmm.
The phospholipids can move around. I was wondering, yes. since the phospholipids tend to move around and move the proteins with them, why don't the proteins end up clumping together in one area? Since it's more energetically expensive, I would assume, for the phospholipids to move around the proteins than to move around each other.
So you're saying how are the phospholipids and the proteins moving relative to each other?
Yeah, I'm kind of asking
So
Why
the way to
is it?
think about it, one way to think about it is the proteins are kind of a raft floating around in a sea of phospholipids. That's kind of a simplistic way of describing the arrangement of all of those different components.
Mhm.
So thinking that the phospholipids is all of the water molecules in the sea moving around, and the raft of proteins is kind of floating within that sea of phospholipids.
Yeah, because like if you like throw a bunch of leaves or rafts into water and just have the water be sloshing around, I would assume that they would all eventually clump together in one
Oh, I
area.
see what you're saying. Why don't the proteins clump to each other?
Yeah.
Well, for for one reason is because they really are not, because the proteins do have a transmembrane domain that is hydrophobic, that is that does form hydrophobic interactions with the lipid fatty acids.
Okay.
All right, so there is a hydrophobic domain to the proteins, right? The part that's actually, if we looked at this guy, the part that's actually interacting, that's why it's held in place, right?
Mhm.
There would be hydrophobic domains, and then what's on the outside would be hydrophilic domains. So that, I think that might be one explanation as to why they don't clump. In
Okay.
addition, often these proteins are associated with other proteins, either in the extracellular side or on the
Oh,
cytoplasmic side. So I think a
okay.
combination of those, I'm not sure which one has it's going to be some combination of all of
Right.
those interactions. And again, that's a good question, because often we talk about just one thing, right? We're very reductionist when we're talking about, especially in this simplified picture, right? You have
Mhm.
one protein and it's interacting with two layers, but everything's moving all the time, and it's
Yeah.
actually very dense, molecularly dense, both the outside, the inside of the cell, the membrane. So there's a lot of different types of interactions that are going on, both
I see.
repelling, right, attractions, and repelling forces as well.
Okay. I
That's a
see.
good observation, though.
Thank you.
Okay. All right.
Okay, thank you so much.
Okay. All right. See you guys later.